You sit down at your desk intending to tackle a difficult assignment, but 10 minutes later, you suddenly find your thumb locked into an unconscious scrolling pattern on your phone. It is common to assume that this gap between intention and action indicates a personal failure of willpower. In reality, the brain is simply trying to survive. It consumes significant energy, and to protect those reserves, it seeks to reduce the heavy lifting done by the prefrontal cortex. The prefrontal cortex is the region responsible for complex, conscious decision-making and active focus. When the brain identifies an action you repeat frequently in the same context, it strips away the conscious thought required to perform it. It converts those steps into a closed, automatic routine. Habits function as a biological survival mechanism, evolved specifically to minimize cognitive effort by offloading repetitive tasks. To automate these behaviors, the brain relies on a highly specific three-part mechanical pathway. First is the cue. This is an external environmental signal or internal feeling that triggers the brain to initiate a specific behavior. Next is the routine. This is the physical or mental action that you execute immediately following that trigger. Finally, there is the reward. This is the biological or psychological payoff that tells your brain this sequence is worth repeating. As the loop repeats, the neural circuitry responsible for the action migrates. Responsibility for the behavior moves out of the conscious decision-making center. Activity in the prefrontal cortex decreases, and a deeper neural structure called the basal ganglia takes over. The basal ganglia is a group of structures deep within the brain that help automate repetitive behaviors and motor functions. At this stage, conscious choice is neurologically bypassed. The basal ganglia runs the behavior entirely on autopilot. The speed at which this loop solidifies depends on the type of reward at the end of the sequence and its specific chemical output. The primary chemical driving this reinforcement is dopamine. Dopamine is a neurotransmitter playing a major role in how we process motivation, pleasure, and reward. This chart shows dopamine levels mapped across the habit loop. A clear anomaly emerges in the data. The chemical spike happens before the reward is even received. The largest surge occurs the moment a reward is merely anticipated, right after the cue is recognized. Behaviors like eating junk food or checking notifications require minimal physical effort and deliver an immediate hit of that chemical. Contrast this with activities like intense exercise or studying. These require high energy expenditure, and the rewards are delayed by days or months, resulting in a much weaker initial dopamine response. The brain's biological preference for instant gratification and low energy expenditure makes negative behaviors naturally effective at hijacking the habit loop. These automated sequences are deeply hardwired into the basal ganglia. Because of this physical structure, they cannot be erased or forgotten through effort alone. To eliminate a negative behavior, the overarching loop must be kept intact. Modification requires swapping the variables inside the existing structure. You start by identifying the specific external cue that triggers the behavior. You must keep the reward structure identical at the end of the sequence. The intervention happens in the middle. You are inserting a new behavior directly between the existing trigger and the payoff. You can also manipulate your physical space to prevent the loop from starting. Removing a phone from your desk or hiding junk food eliminates the initial triggers, stopping the sequence before the dopamine anticipation can spike. Lasting behavioral change is a result of engineering the physical environment and hacking the mechanical variables of the brain's automatic loops.